Hi all. Today we're going to do the cell membrane diffusion and osmosis lab. Okay. To start with, I've set up the Brownian movement procedure number 1, so you can see that particles are in movement at all times. And so all I've done is taken a little bit of Indian ink, okay, and placed it on a slide with a cover slip, okay, and tossed it under a microscope. So now what I'm going to show you is through the connection to the camera, to the computer, I'm going to show you the particles in motion, okay, called the Brownian movement. Okay. And so often when we talk about biology and chemistry, we often say, well, particles are always moving. Even the particles in the desk, in the computer, in the camera or the microscope, in our bodies, they're always in movement. Now, the movement might be very minute, but they're still moving. Okay? And this is one way that we can show you that, sure enough, you can see these individual particles of Indian ink and how as the light source, as it increases the temperature of the slide, the movement of these particles should speed up. Okay? And so first, I'm going to get you so you can see the movement, and then hopefully I'll stop the camera, come back in a little bit after the slide's really warmed up, and then hopefully that movement is a little more fast, or at least a little more apparent to you. Okay, so let's get this set up. Alright, so right now it is at, 40 times your eyesight. You're not going to be able to see the molecules very well there. So now I'm going to go 100 times eyesight. And you can see molecules here. You guys won't be able to see anything until I get a little bit closer, and then I'll adjust things. And then I'm going to go to 400 times your eyesight. And now we'll look for that particle movement. Okay, there you can see it. Alright, so that's a pretty good example. All those little dots in there, that is Indian ink particles, and you can see that they're definitely moving, they're colliding with each other, and that's what we call the Brownian movement. So this is nothing more than just Indian ink on a slide, and with some temperature being applied through the light to cause the movement to become more rapid. So I'm going to give it a few minutes under high intensity. I'll turn the camera back on, and we'll look to see what has happened to that Brownian movement, if it's moving more randomly or in other patterns and whatnot. Okay? Okay, so sorry about the reflection before. I guess I didn't really notice that. But now you can see, at least hopefully you can see, that the movement of these particles is a lot faster, actually. And maybe you can't tell from the video, but if you're concentrating on some of the bigger particles and their movement, you can tell that they're really moving now, the bigger particles, which means a lot of the little particles are slamming into them and making them bounce around, and you get a lot more energy from that source. Okay, so hopefully you can see that and understand what Brownian movement is, and that you believe chemists and biologists and physicists when we tell you that all particles at all time are in motion. And it's easy enough to see this from just putting a little ink on a slide and throwing it underneath a microscope. Okay? The next setup I'm going to look at is I'm going to show you diffusion. And so if you remember from lecture, diffusion is the movement of molecules from a high concentration to a low concentration across a membrane. Okay? And so we're going to look at that in just a little bit. Okay, so I'm back. Now I'm going to demo out diffusion and osmosis. So remember, diffusion is movement of molecules from high concentration to a low concentration. Okay? So this could be salt moving from high concentrations of salt to low concentrations of salt. It can be really anything. Okay? Now osmosis is a type of diffusion, except for we differentiate between movement of water and movement of all other solutes. So if water is moving from a concentration of high water to a concentration of lower water, or from a concentration of high water to a concentration of high solutes to try to reach equilibrium, we call that osmosis. Okay? And so that's what I'm going to demo out right now. So I'm going to first show you how I'm making semi-permeable membranes and how I'm kind of making a makeshift cell. So you can think of this as kind of a cell structure or makeshift cell. So I'm going to demo this real quick like. Alright, so right now I have some dialysis tubing that we've cut into sections here. And dialysis tubing, as the name implies, is hollow. It's tubing. So I've tied off one end with a piece of string, and then I open the other end so I can insert some stuff. And to this one I'm going to add 20% starch solution. So I'm going to mix it up and get some starch in there, and then just add that straight to the bag. So about 5 to 10 milliliters. So really how much you use is dependent purely on how big of dialysis tubing you have. But so there you can see I have it in there. I'm just going to kind of give it a little twist, and then I'm going to tie off this end. So do a couple simple overhand knots, get it pretty tight. Now occasionally when you do this, dialysis tubing will rip or break or whatnot, so we'll see what happens. Hopefully my dialysis tubing is fresh enough and good enough that this will work on the first time, so this is not a really long video and I don't have to keep making it over and over again. But I'm just going to rinse that dialysis tubing off so there's no starch on the outside, so we don't get a incomplete reaction or a really fast reaction. Okay, so we're going to jump over to the solutions that I have now. So I have two setups for diffusion and two setups for osmosis. So the first setup for diffusion that I have is going to be this setup here. Inside this dialysis tubing is water and phenolphthalein. Now phenolphthalein is an indicator of a base. Okay? And so what I'm going to do is place this into a bath of sodium hydroxide and water, and we're going to see what's moving. Is the phenolphthalein going to move out, or is the sodium hydroxide going to move in, or are we not going to get any reaction whatsoever? So we're going to place that in, and you already start to see what I was hoping I wouldn't see, and that looks like the bag is leaking, but we'll see what happens. Okay? The next one is starch, which you just saw me fill, and iodine. Remember, iodine is an indicator of starch. When iodine comes in contact with starch, it turns black. And so we're going to toss that one in, and as long as I don't see black rolling out, we should be all right for now. And then I have 2 Osmosis. This one is sucrose inside a dialysis tubing. Okay, so it's just really corn syrup. Okay. And I'm going to toss that into some distilled water. Okay, get it down in there. And then the other one is just a sack of water. Okay, distilled water, and I'm going to toss that on to some sucrose. So there's some corn syrup in the bottom of that. Okay, we're going to give it roughly 30 minutes, and then I'll come back and I'll show you the end results. And I assure you, you can see this one as it is right now. I'm going to redo this one because that bag is leaking, so it might have a hole in it. So I'm going to redo it, and then so you're probably not going to notice all the pink spilling out of that bag. At any rate, I'll bring it back and show you the end results. Well, we are going to finish this one up with procedure number 4, Plasmolysis and Turgor pressure. So first we're going to examine Turgor pressure in normal cells, plant cells in this case. And so underneath the scope right now I have a Elodia leaf, and I know in the lab manual I tell you to use onion. It really makes no difference. You can use any plant cell that you can visualize under a microscope. I just happen to have Elodia in the lab, so that's what I grabbed. So we're going to look at normal Turgor pressure, which means the plasma membrane is full of water. The cytoplasm consists of about 90% or so water, so it's completely full. If the vacuoles exist in large number in the plant, those vacuoles probably will be storing a bunch of water, and we consider this Turgor pressure, so the water pressure inside a cell. To kind of get an idea of how this works, if you've ever cut lettuce or cut celery or something like that, or cucumber or anything like that in a garden and then just left it sitting out in the sun for a few minutes, maybe an hour, and it went from being crisp to limp. Okay, that limpness means that plasmolysis occurred. It means that the water inside the cells is coming out. And that's through transpiration properties, because it's hot and there's no new water coming in. We're going to induce this by adding sodium chloride, adding salt to this slide. Okay? So let's first look at a normal cell. So if I flip you around, this is just a normal Elodia cell. I'm going to, if I can, I'm going to zoom or get into full size here. Okay, so that's a normal Elodia cell. That's under 40 times your eyesight. I'm going to go all the way to 100 times or 400 times your eyesight, so we can start to see these cells a little bit better. So just bear with me as I'm moving through the slide. I'm going to try to find a good region of the slide to examine, so you can see a bunch of chloroplasts and then see this in effect. All right. Okay, so here are some normal plant cells. Let me get to a decent region. Okay, so right in here is pretty good. This plant leaf is a few cells thick, so that's why it looks blurry on one side and then clear on the other. I have to go through a few layers of cells for you to be able to see the layer that I'm looking at. Alright, so here's not too bad. It's also a good example of what we call cytoplasmic streaming. So if you see this region right here, you can see these chloroplasts. So those little balls are the organelle chloroplast. And you can see them moving in the cell. And that's called cytoplasmic streaming. So the cytoplasm, remember, the membranes are a fluid mosaic, but the cytoplasm also moves. And so you can get this rotation method where, you know, these organelles are not staying in one place, they're moving throughout the cell. And when we supply heat, like we're doing right now with the light, sometimes that induces a little more of that streaming action than you would normally see under normal conditions. And you know, it's kind of hard to see. I think most of you can probably see that these are green in color. So those are organelles. Those organelles are chloroplasts. The green is chlorophyll. That's what's reflecting the light there, so you see green. Alright, so you see those normal cells. The plasma membrane is going to be right along the edge of the cell wall. Okay, so I just want you to concentrate on looking at these chloroplasts. Watch these chloroplasts, and I'm going to add sodium chloride, okay, 40%, to this slide and see if we can induce some plasmolysis on the slide. Okay, so get the Turgor pressure to go away and increase and cause plasmolysis to occur. Now, it went out of focus a little bit because I just added sodium chloride to this slide there, but we should be alright. We should be in the same spot. I should be able to get you back to where we were when you're watching that move. Now, if you look right here on the edge of this slide, edge of this cell, you see that cell membrane? That's the plasma membrane. You can see it walking down. What's happening, so is this one, what's happening is I added sodium chloride to the outside, and you could see that the water, you can't see the water move out, but the water's moving out, which is causing this plasma membrane to shrink up. And you can see one here, it's shrinking up, and pretty soon all those chloroplasts are going to be in a great big ball, just kind of like that cell there. All these chloroplasts are going towards the center of that cell, and there you can see it moving more. And that's because all the water, through the process of osmosis, is leaving the cell and trying to reach equilibrium with the salt on the outside. Right? So there's a really good example of the plasma membrane pulling away from the cell. Now you can see plasma membranes in all these cells, where before you couldn't see the plasma membrane. Now you can see it. The cell wall hasn't moved. Remember, the cell wall is made out of cellulose. So the cell wall is stationary, it's not going to move, but the other material in the cell definitely will move as you reduce the amount of water content in that cell. Okay, so hopefully that was pretty clear, and how that works, how plasmolysis works in Turgor pressure. And next time we'll look at cellular respiration and fermentation. So we're back, and I'm going to finish up by giving you the results of the diffusion and osmosis experiment. So first I'm going to collect the results for the very last beaker, the sucrose with a dialysis bag of water in it. So I'm going to try to shake off that extra sucrose, and then we're going to run it over to a scale, and I'm going to weigh it out. Now, I weighed all of them before I put them into the beakers. Okay, and the weight of this one is 10.68. Okay, so again, that's the dialysis bag that had water in it, and it was stuck into sucrose. Okay, so 10.86, I think is what I said. It really makes no difference because the numbers are drastically different. So in the sucrose solution, so the sugar solution, this is osmosis. With the bag that was full of water, the weight of the bag decreased by 3 grams. And so you can see that this bag became more flaccid, less water in it. And if you don't think that's true, if you look at that sucrose, look how runny that is. So this is corn syrup, and you know, it shouldn't be that runny. That's a lot of water in there. So a lot of water leaked out of that bag, came out of that bag. The next one was sucrose in the bag, water in the beaker, and it went from an initial weight of 10.1 grams to 12.83. So it gained weight, and that's pretty easy to see. If you look at that bag, you can see air bubbles in it now. You can see that it's like moving the water. You can actually see water in there. And so it gained water. All right, now for diffusion. Our bag that had iodine on the outside, so iodine and water in the beaker, you can see it looks the same, just orange with iodine, but the bag itself is very different. The bag is black or purplish, okay? And so it gained weight, not very much. It went from 12.5 grams to 13.15 grams, but definitely iodine is an indicator of starch, so you got a definite indication that starch is in that bag, which should tell you which material moved in. Okay? Sodium hydroxide beaker, clear. Okay? I finally got a bag that didn't break. Okay? It went from 8.22 to 8.84, so it gained a little bit. Okay? But remember, phenolphthalein is an indicator of a base. Okay? So there you have it. You can see that that bag turned pink, and those are the results. So based on these results, you should be able to answer the questions on what's the properties that you would expect for movement across a cell membrane, at least these cell membranes, and what color changes you expected, and what happened in the 4 different plasma membranes or semi-permeable membranes. Okay? With that, you should be able to complete the lab.